chapter. And then we're going to go back next week and start reviewing other stuff we've done through the year and introduce what that means in the bigger picture. So let's talk about gene linkage and polyploidy. Crossing over of linked genes is a source of genetic variation. And really that's what this whole section, this whole chapter is about. How to get variation. How can we have so many members of a species and yet each one of them is unique and different? So your objectives for this section are to be able to summarize how the process of meiosis produces genetic recombination. And I really think you probably got a pretty good handle on that. We talked about meiosis, the randomness of it. We talked about fertilization, the randomness of that. We talked about crossing over, increasing variability. So we're going to try to tie that together now in this one. But I think you probably got a good handle that this is all about genetic recombination, which really is a way to say variability. Next thing you're going to need to know is how to explain why gene linkage can be used to create chromosome maps. This one, I think you'll get. It's a little tough, but stick with it. Look at some of the other material I put out there, and you'll get it. And then we're going to lastly talk about something called polyploidy and why that's important for agriculture. So, vocabulary for this. Proteins. Everything we talk about. Traits. Traits are the results of proteins, either enzymes or directly from proteins. Genetic recombination just means new combinations of genetic material. Nobody gets exactly the same piece of DNA as anyone else, with the exception of identical twins. And the third thing is polyploidy. Polyploidy is having an extra entire set of chromosomes. That can happen in plants. It can happen in a few animals, but most of the time it's not good. But when it happens in plants, it turns out to be a good thing, especially if you're a farmer. So again, the more things change, the more they stay the same. How is it possible that we have so many different individuals of a single species? There are close to 6 billion people on this planet, and except for identical twins, no two of them are the same. Everything we talked about in mitosis, everything we talked about at the level of a cell was about limiting change. We didn't want mutations to occur. Meiosis is different than that. Meiosis is all about allowing for change in the form of new combinations of the building blocks that make up that species. So we make an organism. We do that by combining two gametes, two sex cells. A lot of times we call them egg and sperm if we're talking about animals, but if we're talking about plants, it could be pollen and egg. Usually the female sex cell is an egg, but the male cell can be different, and it's not always egg. So this section is all about figuring out, tying together how those instructions are organized and how we can mix and match them to get so many different individuals. So genetic recombination is nothing more than a new combination of genes. Genetic recombination is built into the process of meiosis. Independent assortment. Independent assortment says that the maternal genes don't care where the other paternal genes go, or the other maternal genes, that they're randomly going to cross over, and we're going to get all possible combinations of maternal and paternal genes within a chromosome in a gamete. On top of that, that gamete from one member of the species can fertilize the gamete, any other gamete from another member of the species, opposite sex of the species, because it takes two sets of instructions to make a phenotype. It takes two sets of instructions to make a phenotype. You have to have two sets of those instructions to make a protein, and those proteins will lead to phenotypes. So if we start out with meiosis, we said chromosomes go to the gametes randomly. There are 2 copies of each chromosome, one from mom, one from dad. So if we want to know how many chromosomes there are in a particular species, we could call that N. For humans, it's 23 pairs. For pea plants, it's 7 pairs. There are a few others out there. You can look up how many chromosomes belong to a species. But if we want to figure out how many possible combinations of chromosomes we could get to do that, you take the number 2, because there are 2 copies of each chromosome, and N, which is the total number of chromosomes, and that will give you the number of possible combinations, the number of possible gametes that organism can make. So if an organism had 4 chromosomes, 2 to the 4th power is equal to 16. So if you have 4 chromosomes, that organism can make 16 different gametes. If an organism, like a pea plant, had 7 chromosomes, that organism can make 128 possible gametes. Humans have 23 chromosomes. 2 to the 23rd power is 8,388,608. So each individual human can make 8,388,608 different sperm or eggs if we just look at the chromosomes. If an organism had 52 chromosomes, and some do, there are this many possible combinations, possible gametes that could be made. But it's important to remember that this is if you just look at maternal and paternal chromosome combinations. These numbers ignore crossing over. When you add crossing over, you get even more variation. You're mixing with the genes within an individual organism. So you're not making a maternal chromosome or a paternal chromosome. You're making a chromosome that's got combinations of pieces from each one. So now we have a almost limitless number. It is not purely a maternal chromosome that you inherited from your mother that you're passing on. It's not just a paternal chromosome that you inherited from your father that you're passing on. You're passing on a brand new chromosome that has pieces of the chromosome you inherited from your mother and pieces of the chromosome you inherited from your father. Crossing over, though, doesn't happen randomly. The further apart you are, the more likely there is for crossing over. And that's turned out to be a very important research tool. It allowed people to map chromosomes. Genes are lined up on the chromosome. They're linear arrangement. And the closer 2 genes are, the less likely they are to cross over. And if you look at using different crossings and techniques, you can figure out if they cross over or not. And if they don't cross over all the time, most of the time, then they are close together, and we call that linkage. That linkage looks like a violation of the law of independent assortment. So in this case, because they're close together, they move together. It's unlikely that crossing over will happen between them. Those are all within an organism. So in an organism, you're making gametes. You could make 2 times 2 to the power of the number of chromosomes you have, different gametes, if you just look at the maternal and paternal chromosomes. Add crossing over to that, and you get an almost limitless number of combinations of new gametes that you can make, with the exception of gene linkage that we'll look at a little more closely in the Chalk Talk. Those are all within one organism. That one organism has to find another organism that has done the same thing to make the other kind of gamete. So if a male has to find a female gamete has to find a male gamete, They've both gone through all these processes, meiosis, crossing over. Now there are hundreds of sperm. One egg is chosen from hundreds of eggs each cycle. So the chances of two gametes coming together is also random. If we look just at a pea plant and we looked at without crossing over, there are 128 male gametes that can be made in that pea plant. And in another pea plant, there are 128 female gametes that could be made. And we get that from the math 2 to the n. Peas have 7 chromosomes. Now, to figure out how many possible combinations there are of that, you have to take 128 times 128, which means there are 16,384 possible combinations that you could get by randomly fertilizing one pea plant with another pea plant. So think about it this way. What is the chance of identical twins occurring from two different pregnancies with the same parents? That would be 8.4 million, rounded off, times 8.4 million, which is 70.6 million. So 1 in 70.6 million could be an identical match to another. Again, if you ignore crossing over. Crossing over says that's not going to happen because now you don't really have 8.4 million. You have billions and billions. You'd have to know how many genes there are. And if you take 2 and raise it to the number of genes that are out there, you're going to get a huge number. But again, it's all about genetic variation. That's the take-home message from this. Meiosis allows for the independent assortment, which says you're going to make all possible gametes you can. All possible combinations of the 2 chromosomes you have for each chromosome that make up that organism. Within that possibility, you also have to add crossing over. Crossing over gives us even more variation within an organism, and it gives us a valuable research tool. It provides us a way to map genes so we know which genes are close together and we can even figure out which chromosomes they're on. And on top of all that, we have a random meeting of gametes between 2 different organisms. So this is how we get genetic variation. Meiosis combined with fertilization gives us the ability to make 7 billion different individuals of the same species. No 2 are alike. All right, now on a kind of side note, there is something out there called polyploidy. And in polyploidy, you get an entire extra set of chromosomes. So instead of having 2 copies of chromosome number 1, the organisms that are polyploidy would have 3 copies of chromosome number 1 and all the others, depending on what they were. If we're talking about pea plants, you would have 3 copies of 1 through 7. This is very rare in animals. Most animals, it is lethal. If fertilization occurred with polyploidy stem cells, those stem cells would not be able to divide. That's always the case in humans. Nobody has 3 copies of all their chromosomes. There are some animals that will do it. There are some goldfish that will do that, and there are some earthworms that will do that. But it turns out that it's more common in plants. Plants don't seem to have as much trouble with extra sets of chromosomes. Oats, wheat, sugarcane have extra sets of chromosomes, and usually in a plant, when you have an extra set of chromosomes, you get a bigger plant and it grows better. If you've ever bought strawberries in the grocery store, you notice they are huge. If you go out to the side of the road in the early summer and pick strawberries, wild strawberries, you'll see that they are very, very tiny, about the size of a thumbnail. And the reason for that is these strawberries you buy in the grocery store have extra sets of chromosomes. They are mutant. They are polyploidy. So genetic recombination involves both crossing over and independent assortment. Early chromosome maps created using linkage of genes on a chromosome. Polyploidy plants are selected by plant growers for their desirable characteristics. All right,